Okay, now this is once again the last part of Watson and Crick's DNA structure. Now when we were discussing about Watson and Crick and Rosalind Franklin, I forgot to mention that for their work, Watson and Crick, along with another scientist, they got the Nobel Prize in 1962. 1962 they got a Nobel Prize. Okay. Whereas Rosalind Franklin did not get it because she died in 1958 because she developed this cancer because of exposure to X-rays. But the sad thing is, these scientists, Watson and Crick and the other one, they never even mentioned the work of Rosalind Franklin. Now, without her photo 51, they would have struggled to, form their actual double helix structure. Okay, so that's the reason why I said they're not related to such. Okay, so now we go to the structure of DNA. Here we are, which is anti-parallel, meaning it has this oxygen. Each of these sugar penters have oxygen on the opposite end. Okay, this is the 1 prime, 2 prime, 3 prime, 4 prime, and the 5th prime, which is attached to the phosphate group. Okay, and always remember the nitrogenous bases are fixed, or it's connected to the 1 prime carbon. Okay, 1 prime carbon. It's counted that way, and the 5th one here, because that's upside down. Okay. Let's move on to this side. Nitrogenous bases. There are 4 different nitrogenous bases. Actually, 5 when we consider RNA. Thymine, cytosine, uracil, adenine, and guanine. Okay. These are the 5 different nitrogenous bases. These nitrogenous bases are categorized under 2 different headings known as pyrimidines and purines. These are all things that we've done in class 11. Just bring it back here. Thymine, cytosine, uracil, they are pyrimidines. Important thing to remember here is they have a single carbon ring. Okay, of course it has nitrogen. They have 3 nitrogens in that ring. But the thing to remember is these 3 have single rings, and they're called pyrimidines. And purines, adenine and guanine, they have double rings. So that's the thing to remember. Okay, purines, 2 rings, pyrimidines, single ring. Okay, and another thing to remember, cytosine always attaches itself, or it aligns itself with guanine, and adenine always goes with thymine. So I gave you an example to remember. Cars in a garage, CG. Cars in a garage. And apples on trees, A always goes with T. Okay. Now we were talking about hydrogen bonds in between nitrogenous bases. There's always a free hydrogen bond between C and G, cytosine and guanine, and there's a double hydrogen bond in between adenine and thymine. Okay. So we'll bring this here. We've got C and G combination. They are combined, or they are attached by 3 hydrogen bonds. And here again, we've got E and T, that's just 2 hydrogen bonds. Cg, 3 hydrogen bonds. Cg again, 3 hydrogen bonds. E and T, 2 hydrogen bonds. So this is how they're fixed. Okay? And if you look at this structure here, on one side it has a pyrimidine, a single ring, and it tells us that on the other side it will always have a pyrimidine. Okay, you cannot have, it will always have a purine on the other side with double ring. You cannot have a purine with a purine, because that is going to bulge the size of this structure as it goes parallel here, and it bulges like that. As if it looks as if a snake had swallowed an egg, kind of. Okay, but in order to prevent that from occurring, you have double rings here on one side, single ring on the other side. Single ring on one side, double ring on the other side. So this maintains its structure. Another thing about double helix, why is it called double helix? We understand the word double, it talks about the two strands. One strand here, the other strand there. Okay? So this strand we just said it is made up of sugar and phosphate. Okay? It's also called nucleoside. So there are two nucleosides. Okay? That's the reason why we call it double. Why is it helix? When the carbon and when one nucleotide joins to another, it kind of, let me give you this example. Let's say this is the shape of one nucleotide, and when another nucleotide joins, it forms a structure like that. When another one joins, okay, it has a narrower end this side, broader on the other side. So what's actually happening to my structure, this chain, is actually it is getting twisted just because of the shape of it. When you fix another nucleotide, it's going to twist again. Okay? So because of the structure, this chain begins to twist and coil, okay, into a helical shape. So that's the reason why it's called helix, double helix. It's not the name of our, oh, of course, it is the name of our science club. Right, okay. Hydrogen bonds. Then the next thing, three codons. Codons are sequences of three nitrogen bases or nitrogenous bases which codes for one amino acid. So codons are a sequence of three nitrogenous bases which codes for one amino acid. Why is this important? Because codons makes amino acids. The amino acid in turn, when they join and form a long chain, produces a particular type of protein. And without protein, none of us are going to survive. Okay, just to conclude this, whatever we've done, what was this? What is this? This is, some of you may be saying things which I can't comprehend, but let me tell you, this is just a piece of football, man. What are you thinking? Just swing your legs, okay? Right, see you in the next video.